bit. So thank you very much for coming tonight. Next week, I will be here, and I will be the speaker, and I will be talking about the Xenos paradox and the futures of science outreach. This is Xenos is X-E-N-O-S. It's a pun, a play on the word Zeno, Z-E-N-O, who had a paradox. And this Xenos means, it's a Greek word that can mean either stranger, visitor, or guest. And the Xenos paradox is, how do we as a university look at people coming to visit here, especially for science? Do we look at them as visitors, or strangers to fear, visitors to accommodate, or guests to welcome and engage? On February 1st, in 2 weeks, Lee Sanderwitz will be here from Gender and Women's Studies. She's a new assistant professor, and the title of her talk is, I Was Obligated to Accept: Coercion and Autonomy in Contemporary Global Family Planning. So I'm looking forward to that. My wife is a sociologist and demographer, and I'm always delighted to include the full range of science at Wednesday night at the lab, especially the social sciences and in particular demography. On February 8th, we'll have a special Darwin Days presentation, because when is Charles Darwin's birthday? 12. February 12th. What year? 1809. And who was born the same day? Way to go. Yeah, I know.
I've had this class before.
Every year I say the same. Okay. Oh, no, this is great. No. 5 down and 7 billion to go. Oh, it's 8 billion now, isn't it? 8 billion. On February 8th, our special presenter will be John Hawks of Anthropology. He will be here to talk with us about finding other ancient minds across the human evolutionary tree. So I'm delighted. He's one of the finest speakers any university has, including this one, and I'm thrilled that he's going to be the Darwin Days presenter. And this is going to be a joint production with the J.F. Crow Center for the Study of Evolution, as it is every year around Darwin Days. And then Marcus, would you like to stand here and I'll do the formal introduction? Yay. This is where we count to 10,000 slowly, and then we have a cut point.
All right.
900, good. Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension, Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to introduce to you Marcus Vinje. He's with the USDA's Agricultural Research Service in the Cereal Crops Research Unit here on the campus of UW-Madison. He's going to be speaking with us about malting barley research. Marcus, I get to ask you the same questions, 5 questions I ask everybody. The standard line is, the responses don't have to be true, they just have to be believable. Marcus, where were you born?
I'm Madison, Wisconsin. I'm a townie.
All right. And where'd you go to high school?
Madison Memorial.
Way to go. And then...
Yeah, it's Madison Memorial now. They just changed their name recently.
Velma Hamilton.
I think it's still Memorial.
No.
You're right,
yeah.
Velma Hamilton, I think.
No, because Hamilton's the middle school. Anyway, We'll figure that out. It used to be Madison Memorial.
It did.
And then where'd you go for your undergrad, and what did you study?
Well, I'll have to call out some UW-Whitewater. So I went to Whitewater for 4 years, majored in hockey, transferred to Madison, and got my degree in genetics. You're killing my first slide here. No,
I'm sorry.
No, I'm teasing.
That's all right.
Yeah, I went.
So what was your major in hockey?
That's it. Defense.
You played defense.
Yeah,
okay.
Checking and good slap shots.
Checking and savings, yes.
Yeah.
See, there's checking for and then
there you go.
And then where'd you go for your advanced degrees, and what are they in?
I went to my advanced degree at UW-Madison as well, and I got it in agronomy. And I was with Stan Duke and Cynthia Henson.
Good.
Stan was the chair at the agronomy department for 11 years, and right when I started, he stepped down, so he was one of my PIs, and then Cynthia was agronomy as well.
Good.
And USDA member.
And then when did you start at the Cereal Crops Research Unit?
2012. which
Right.
feels like a long time ago when you say it out loud, but feels, we've been there for a little bit. I'm kind of the old hat there, as some of our new people know. Like, there aren't many of us old hats left at CCRU.
I bet they're using a different word than hat.
Yeah.
Well, I'm looking forward to this. This is one of the most fascinating biochemistry and agronomy talks, two things that I am fond of, plus you end up with beer.
Beer. That's the best part about it. No barley, no beer.
And Horlick malted milk for those of us who can go way back in Racine.
There you go.
So would you please join me in welcoming Marcus Vinje to Wednesday Night at the Lab.
Thank you, Tom. What Tom did not mention was how he got me here tonight. So my daughter is a biochem major at Madison, and she was getting picked up by my sister. Like I said, I'm from Madison, so she was picking up my daughter on campus, and her car broke down. It happened to break down right in front of Henry Mall, right in front of the Biotech Center, and obviously I'm right down the street, so they call me to come help, and as I'm helping the car, this car is dead. The axle fell off, so it's not movable. We're switching stuff to my car, and Tom's helping us out, putting up signs, and somehow he finds out that, I have a lab down the way, and I'm still loading stuff. He's like, "Hey, you want to talk?" And serendipity has it, here I am. So really excited to give you guys a little information on what we do down at the Cereal Crops Research Unit. So with that, I will get started. All right, so a little bit, this outline of this talk, I'll talk a little bit about me. We kind of covered that earlier, so that'll be faster. I kind of want to talk about barley and in relationship to Wisconsin as well as UW-Madison, and that'll kind of segue into the history of the CCRU. And then I kind of want to give you guys a lowdown on what we do down at the Cereal Crops Research Unit, and talk to you guys about the researchers there as well, and then end the talk with the last 35 minutes on what I do and some of the stuff I'm interested in. Like I mentioned, I'm a townie, so 04, 09, I got my degrees from Madison. Grew up in Madison. My grandfather was actually a professor at UW-Madison. He was in the agronomy department, and so in 2007, my first conference was Tri-Societies. So my grandpa was a member of the Agronomy Society. He was a fellow. He was down there at the 100-year anniversary to talk about the history of the Agronomy Society, and I was presenting for the Crop Science Society. So we had a pretty fun time down in New Orleans during my first conference. All right, so let's talk about barley at UW-Madison. So in the late 1890s, they really started to look at improving barley at Madison. They started looking at variety performance. So one of the better varieties we had back then was Oderbrucker, is a German variety, and then the other one that was kind of prevalent at that time was called Manchuria, and those are the only two kind of being used for malting and brewing. They weren't really focusing on malt quality back then so much as looking at disease resistance, because these were kind of susceptible to disease. And then in 1920, we had prohibition, so they definitely weren't looking into malt quality, but they were, yeah, exactly. They were breeding more for feed attributes, yield and smooth-ons. So in around 1900s, malt was still important in Madison. So one of my scientists at the Cereal Crops showed me this book that showed that there's a malt house on Lake Mendota around 1900. So this is the malt house. It's no longer there at all, but that's just how close we were to malt back then. And this is a picture of Ransom Moore. So this guy right here is the namesake of the agronomy building. So it's called Moore Hall, and that's Ransom Moore. And he's standing in a field of Oderbrucker barley on the west side of Madison. I think this is kind of where the hospital is, but it's kind of similar to where we are, the Cereal Crops at right now. And this was the cover of the agronomy history book. So barley and UW-Madison has a long history. So at the end of Prohibition, we started looking at malt quality at UW. Some simple traits here, and then diastatic power. We'll talk about that a little more later on. The problem is Wisconsin didn't have the best environment for growing barley. It's a little hot and humid during harvest time, so that can lead to disease, as well as pre-harvest sprout, or essentially germinating on the plant. So Madison's history of releasing barley varieties was strictly feed and forage for the last 50 years. And that kind of culminated then in UW-Madison stopping their breeding program. However, that was in '01. Since then, Dr. Gutierrez has come on board, and she is a geneticist and has a population of barley that she's working on, as well as wheat and oat. And then Dr. Conley, he primarily focuses on soybean, I believe, but he's got a small greens project going on and looking at growing barley in local environments, and we're actually malting some of his stuff right now. Okay, so end of Prohibition, 1933, the Barley Malt Lab was established. So this was started in the basement of the Agronomy building, and they were kind of looking at doing some performance testing and helping out all the growers and the maltsters around town. In 1940, they began evaluating breeder selections from state and federal experiment stations, and that's something that we do to this day. So this malt lab was being used quite a lot, so much so that they decided they needed their own building. So in 1949, they opened a nice new building at 501 Walnut Street, which is kind of by the cogen plants right now. In 1983, we merged with the Malt Lab and the Oat Quality Lab merged together to create the Cereal Crops Research Unit, and that's what we're called today. However, a caveat, our last oat researcher retired in 2017, so right now we're strictly barley and malt, which our stakeholders are happy about. All right, so here's what the fancy Barley Malt Lab looked in 1949. Nice fancy equipment. It was the state of the art. They did a little brewing and mashing as well. We don't really do that so much anymore. This is a picture from across the street. And in 2006, we moved from this building to our new facility, and this is down at 502 Walnut Street. So if you see, this is right down here is the old Barley Malt Lab. It's now just an empty lot, but Google Maps still has it labeled as the Barley Malt Laboratory. And here, we're located right here. I pulled this off like a month ago, too, so not too updated, but it's kind of nice to see that next to each other. So in this building, we have 6 research labs, currently housed by 4 PIs, as well as a malt quality lab. So this whole region facing our parking lot, that's our malt quality lab. Everything on Walnut is our research laboratories. Here's where the scientists sit. So I'm going to talk a little bit about the Barley Malt Lab right now and what they do in their process. So like I said, we get a lot of breeders' lines in. First thing we got to do is we got to sort them. We got to count the seeds and get a grain weight, and then we do something called plump, right? So you don't want to malt thin seeds, so we focus, so they run through these screens to get plumps, and once you got all these processed, then we malt them. So our procedure, this is a steep tank, so you put your barley in these little cans, they can hang on this little hook, and they get dunked in water for 4 hours, and then they get a rest in the air for 4 hours. They repeat that until we get about 45% humidity. Once they reach that stage, we take them to the germination chambers, where you keep them cool, you keep them humid, and you rotate them every once in a while. Here's what it looks like on the inside. So these little cans will hold your germinating barley. Then after that, you heat it up in an oven. We call it a kiln. And all of this process is essentially trying to mimic industry scale malting. So RAR is one of the biggest maltsters in the country, if not world. So these huge steep tanks where they're steeping their barley, then these large, these are as big as football fields, just huge floor malting facilities, kill them in these large facilities, and then store it. So we take this large scale, we try to make it smaller, but still comparable to what they do in industry.
And where are they located?
That's in RAR. I think this is in Shakopee, Minnesota. I could be wrong. Sorry, RAR, if I'm wrong, but I know that I think it's in Minnesota. They like to be near where they grow the barley, and that's the northern plains states. So once we get our malt, then we need to analyze it for the malting quality parameters, because we're doing this analysis for the breeders. They need to select which lines have the quality attributes they're looking for. So this is where the magic happens there. We have a lot of specialized pieces of equipment. You got to grind your barley to make beer. So got grinders. You got these are small micro mashes. So when you make beer, you go from malt to mashing, which gets you your sweet worts. This is what that's doing. Then we have some of these heavy lifters, like this scalar. This scalar, it's able to run a whole bunch of different assays and get a whole bunch of different numbers for the breeders. Now, I mentioned about malt quality a bunch, and I haven't really talked about what malt quality is, so we'll go a little bit into about what is considered malt quality. Essentially, it's a complex genetic trait that's determined by measuring numerous parameters. And these are just kind of like the headings of the parameters, right? So you got barley factors, that's just raw barley. Then what happens once you malt it? Then you got measures of how well that malt has been modified, and et cetera. So here's the next picture is a picture of 7 barley chromosomes and where all the barley malt or the malting QTLs have been found, as well as enzymes. So you can see that it's kind of a whole genome thing. All of these are associated with malt quality. So it's a somewhat convoluted, especially considering the interplay between some of these parameters. So I work on diastatic power a lot, on the enzymes that produce that. And the problem we have with that is grain protein, we want that at a minimal level. But when you get high grain protein, you get high diastatic power. A lot of brewers and maltsters want high diastatic power, but they don't want high grain protein. So that relationship is confounding and can be tough to work with. The American Malting Barley Association is our main stakeholder. They get together with the industry folks and they determine what they want out of an ideal malt. So the breeders use these guidelines to target for their breeding lines. So if you see diastatic power, it's down here. You know, if you're brewing a Miller Lite, you want it above 140. If you're brewing Fantasy Factory, you want it a little lower. But if you're brewing whiskey, you want high DP. All right, so when we look at the malting quality analysis at the cereal crops, so all these breeders in the UW, or if you're associated with AMBA, our trade association, you can have access to our laboratory. There are 6 full-time employees that are just working on this malt quality. They were able to go through about 5,500 to 6,000 samples a year. And I think as of right now, we got about 83,000 samples run since we started doing this for the public. And when they look at all these factors, so when you get a report from our lab, you will have data on all of these parameters. So here's, like I said, the malt modification. They'll look for the beta-glucan levels, which is the cell wall breakdown, as well as like a soluble over total protein. All right, so that's our malt quality lab. So that's like one half of our building. The other half of our building is research. And here are our 4 researchers. So they got me over here. I started in 2012. Dr. Mahalingam, Molly, he's our research leader now. He started in 2015. The director of the Malt Quality Lab, as well as runs his own research facility, is Jason Walling. And then we have our newest hire, who's a research biochemist, and that's Sarah Whitcomb. We're very excited to have her. And the next few slides, I'll kind of give you a brief overview of what the other 3 scientists are up to. So Molly is an expert in heat and drought stress. One of the first things he found when he was doing research here is that when you put barley plants under heat and drought stress at the same time, if you do it early, when it's just in the vegetative stage, the barley is able to recover and still get malt quality. Now the problem is if your barley in the field doesn't make malt quality, then that grower is not going to want to do it again, right? He's not going to get the same premium he gets from malt if it doesn't meet those parameters. And what he found is if it get heat drought stress when the barley just starting to head, that can completely ruin the malt quality. And that's detrimental because especially with climate change, it's getting warmer, it's getting hotter, it's getting drier in certain areas, so we need to have barley that's more adaptable. So that's what he said here. He has identified 3 accessions that are tolerant to the combined heat and drought stress in the mini core population that he's working with. Another research goal he has was identifying the important proteins that develop during malting. So he sequenced the proteome of the malting barley as well. He's also done work on toco biosynthesis pathway. He's identified some SNPs using GWAS And then he's also looked at this BRBO gene family. Some of these may be a proxy for germination that could be used in malting. Dr. Jason Walling is the director of the Malting Quality Lab, as well as runs his own research program. One of the main things that he is interested in is the genetics of beta-glucan accumulation, as well as pre-harvest sprouting. And what you here see is a panel of different levels of pre-harvest sprout. So like when the barley is on the head, if it reaches certain conditions, it can start sprouting, as you can see right here. And he identified 3 genes and some mutations within those genes that can predict pre-harvest sprouting. Another research goal he's looking into is using hyperspectral imaging to predict when the seeds are germinating, and then with the hopes of taking that to the field and being able to identify that in real time. Our newest scientist, who we're very excited to work with, is Sarah Whitcomb. She started a few months ago, and her main research goals will be looking at the metabolome. She'll be looking at the malting metabolome first, but then she'll also look at the dry barley metabolome as well as what's in the wort. So when the barley is mashed into to the wort, that's what the yeasts are going to eat. So there's a lot of interesting compounds in there that she's going to be working on. So we're excited to have her in our lab, especially considering that we have this really cool everybody in our unit project where we're trying to integrate multiple omics on malting barley. So in 2020, I published the transcriptome of malting barley. Molly published the proteome in 2018, and Jason and I sequenced the small RNAome a few years ago, and Sarah's currently analyzing that right now. And then with the combination of the metabolome, we'll have all of these omes, and we're hoping to identify some functionally characterize some key genes that are important for malt quality malt quality traits. So now that is what our building does besides what I do. So from here on out, I will be speaking about my lab. So my lab, well, this is kind of like my mission statement for my lab. I'm really interested in understanding the regulation and expression of genes that are associated with malting quality traits. I'm really looking into genes that encode for enzymes that can attribute to the fermentable sugar production and malting quality trait diastatic power. I'm really looking forward to identifying new genes that could influence these traits, specifically diastatic power, and looking into some of these minor carbohydrases and some of the genes that I've recently identified. I want to make sure that they create functional enzymes that can actually break down starch. So we mentioned diastatic power a few times, so I'm going to give you a little background into diastatic power and some of my research background before we start getting into the actual research. So diastatic power is essentially the ability of malt to degrade starch and make fermentable sugars. So this is a picture of an alpha-glucan chain or starch, and there are 4 main enzymes that are able to break down the starch. Alpha-amylase is a big one. You can see alpha-amylase right here. It's cutting these alpha-1,4 glucosidic bonds, and it's an endoamylase, so it can go inside the chain, and it can also work on the starch grain itself. The beta-amylase, so I'm partial to beta-amylase. I work with it a lot. You'll hear some more about that one. Beta-amylase releases beta-maltose. So it releases maltose, which is just 2 glucose residues combined, and it breaks the same bond, but it can only work on the outside, so it's an exohydrolase. Alpha-glucosidase releases glucose. It can also break down maltose into 2 glucose units, as well as break the inside chain, just any kind of alpha, or it can go into anywhere and just break off 1 glucan chain. Limit dextranase is the enzyme that breaks these 1,6 bonds. So in amylopectin, which is a form of starch that has these 1,6 branch points, the other 3 enzymes cannot get past certain if there's a branch point, so you need limit dextranase to come release that, so the other enzymes can continue to break down starch. All right, so I mentioned beta-amylase. That's my favorite one. It's the main driver for diastatic power. So what you're going to look at here is, so these are enzyme activities correlated to diastatic power. Beta and alpha are both involved with diastatic power. Beta, however, seems to correlate better in all the studies that I've seen. sometimes perfectly. All right, so maltose is the most abundant sugar in malt. So beta-amylase produces maltose, and if you look at the sugar levels in just straight malt, so this is as malting goes on, so that's right out of steep, and then 24 hours later, and that's at the end of malt, you see maltose is by far the highest sugar, 60-some% at the end of malt. Now when you take that malt and you grind it up and you add water to it, you're mashing it, you see that the sugar, the predominant sugar, is also maltose. Maltose ramps up and ends up staying up here, but it's much, much higher than all the other sugars. Probably why they call it malt, huh? So, all right, so a little bit about my research now. So when I mentioned I'm homegrown, I did my research here at the U at Agronomy for a member that for USDA and a UW professor. So in 04-09, I was studying the expression of two barley beta-amylase genes. At that time, those were the only two genes we knew about in barley for being beta-amylases. They were both functional enzymes. I was looking into markers to predict enzyme or activity as well as thermostability, and we were tipped off that there's this third intron in beta-amylase that's pretty that there's this indel within that region that can predict enzyme activity and thermostability. So I went in and sequenced a whole bunch of wild and cultivated barleys, identified 4 haplotypes, and then we started screening our germplasm for that. And what we found is in the North American germplasm, we didn't get much usefulness in prediction. However, these markers or these haplotypes have been used in Australia as well as in Asia. I just reviewed a paper last year that showed that one of these haplotypes conferred high activity in one line. So it's a good marker, but not so much for our germplasm here. So when I started postdocing in 09 to 12, I still was interested in that intron 3 haplotypes. So we did a little more work trying to find if like malting or mashing sugars can correlate to that, but we didn't really find much utilization there still. I went in and looked at some BAME2, the second gene, some of the regulation with that. And then one of my favorite things that I did was look at this beta-amylase gene regulation. I've tried to put more of this into my talk, but I couldn't get 50 minutes in there. But essentially what I did was I attached the BAME1 promoter to green fluorescent protein, and I shot it into barley, and I kind of wanted to find where the critical regions of the promoter was. We identified 500 base pairs upstream of the start site that was essentially conserved in wild barley as well as malting barley. And we identified these cis motifs that were very important. And then we got cool glowing barley pictures. All right, so now I got my job. I'm a research geneticist at the USDA. I'm still interested in BAME1 regulation. I did a research where I found that hordeins are the main seed storage proteins in barley. You might have heard of them as another name, gluten. That's the equivalent of gluten in wheat. So I studied the expression of both of these two, because as I mentioned, that green protein and diastatic power conundrum that I mentioned earlier. I was trying to see if maybe hordeins were expressed at a little later time during development. Turns out they're expressed at the exact same time. So here's BAME1 peaking at 25 days. HOR1, which is the B hordeins, looks the exact same. C hordein looks very similar. So they're co-expressed. Now, why did I mention that? Well, there is this mutant called the RESO 1508 that has this lyse3A phenotype and has very low hordein and beta-amylase gene expression. So they created this mutant back before I was born, but it's been used throughout this time for the last 40, 50 years. And what they were looking for was high lysine. High lysine is important if barley is being fed to animals. Lysine is an essential amino acid, so that's essentially what they were looking for is for feed. And they found one that was really high in lysine, and it also knocked down hordein levels. And it also knocked down, I did some graphics. So if you look at here, this is a Western blot, and this is RESO 1508, the mutant. 1 and 6 is the wild type, and you can see it's missing large bands. Right? Down here is beta-amylase, and you see in the mutant, you see a big blank spot. So we know that this mutant affects both of these. So we wanted to looked at further, especially considering of this paper. So they, he, identified that in this mutation, or in this mutant, this lyse-3 mutation, the B. hordeum genes did not have accurate methylation. So in the wild-type endosperm, these open lollipops indicate there's no methylation. So in the wild-type endosperm, they needed to have the methylation removed, whereas in the mutant lyse-3A endosperm, you see hypermethylation or methylated. They did not find that in D. hordeans, so this wasn't for all the seed storage proteins, but they did, so the D. hordeans did not follow this. So nothing was methylated in the D. hordeans, but the B. hordeans were affected. In a conversation with Crispin Howitt, who used this RISO 1508 line to essentially create a gluten-free barley, these Australian scientists, in talking with Crispin, who gave us some of his lines to work on, he mentioned that this group thinks that this lyse-3A phenotype is a mutation in the gene Demeter. Demeter is a demethylase, so it will remove methylation from promoters in early endosperm development. So I was interested to know if this phenotype is actually caused by mutation in Demeter. I also wanted to look at if the BAME1 promoter required this demethylation to have gene expression, and then I kind of wanted to know just what genes were being differentially regulated in these mutants. So I identified 3 different lyse-3A mutants in 3 different backgrounds. So this is the original mutant. I call them Bowman lyse-3A just so you know the parent and you know the phenotype. Then Bowman lyse-3A takes a feed barley, bred it with 1508, and then back-crossed it 6 times. And then this one was from that group that created the gluten-free barley line. So we had 3 backgrounds for the lyse-3A, and then we had these 2 allelic versions that were created around the same time as this one, but had the same phenotype. And then we had their parents. And a lot of this methylation work was done by Carl Simmons in my lab. He was great. So we looked at 21 days after enthesis endosperms. We got the transcriptome, we did the methylome, and then we did some gene and enzyme activity as well as gene expression. So our goal was to determine if BAME1 required demethylation, and then to just determine what genes were being upregulated and downregulated. So originally we wanted to look at just beta-amylase, just the promoter beta-amylase. However, we got all our primers ready, and then the pandemic hit. So it took us a while to get back into the lab. Didn't really have much time to troubleshoot because it was tough to get in there, so we decided to just look at it all, and we did the whole genome bisulfite sequencing. So we looked at total global DNA levels, and this is what we got. So 3 columns, each represent a different methylation point in the DNA, so CG, CHG, CHH. So CG, so obviously the first thing we all see is this one right here. That we think is an artifact. We think that one of the reps just did not sequence well. So if you just ignore those and you look at Bowman versus the mutant, you don't see any difference. If you look at Bowman versus Bowman lyse-3A, you don't see too much difference. Nothing that really stuck out at us. So from here, we're like, okay, well, let's look at some high-expressing seed storage protein genes as well as beta-amylase. So Carl wrote some different code to be able to pull out 1,000 base pairs upstream of every start site of the genes that I gave him. So what we see here is these are very highly expressed genes. This is what they're colloquially called. And we also have beta-amylase 1 here. So that paper that showed that B. hordeans required methylation, demethylation, we think they might have been looking at this one gene. As you can see, there's a lot more B. hordean genes than they looked at. But we think that they might have been looking at this, because you can see in the wild-type Bowman, no methylation. In the lyse-3A, you see more methylation. And here you see this lowering level of gene expression. However, if you look at SLOOP and SLOOP lyse-3A, you see 100% methylation, and you still see a reduction in expression. So that didn't really follow that like we anticipated. Especially, and then if you look at this gene, which is 1420 right here, similar methylation between Bowman and Bowman lyse-3A, no difference significantly, but look at 135,000 to 1,000 TPM. So huge reduction did not seem to be caused by any methylation difference in the promoter region. And like I mentioned, we were interested in BAMI1. Here's BAMI1. BAMI1 has 20% methylation in the wild type, 7, so less methylation in the mutant. However, we see a large drastic reduction. So it didn't seem to be causing the methylation issues that we predicted based on that 1992 PNAS paper. All right, so now we're going to switch. So we looked at some of the methylation. Now we're looking at some of the differentially expressed genes. So to get this Venn diagram, you took BOMI versus BOMI-Lys 3A. You get a set of differentially expressed genes, and then you compare that number to all the other mutants and their parents to get a Venn diagram where we got 29 differentially expressed genes that were common among all mutants and parents. So if you look at these numbers, so these are the highest expression. Only 10 really had high expression. So differentially expressed genes, sometimes they can be considered differentially expressed, but they could be really low expression. So I wanted to look at the higher expressed ones. And what you see is that, yeah, you see some of these seed storage proteins, they're down-regulated quite a lot. So look at SLOOP, and they're down-regulated. That's what we expected. One thing that we found that was interesting was this beta-glucosidase. So it gets upregulated in all these mutants, right? Well, why is that exciting? Because these mutants are notoriously low in beta-glucan. So here's our malting cultivar. It gets about 147. If you look at these ULG numbers, which have that Lys 3A phenotype within them, they have very low beta-glucan. So we predict that that's caused by that increase in beta-glucosidase. So now if we're just looking at our Lys 3A, so comparing just the Lys 3A phenotypes in the 3 backgrounds, we got 118 differentially expressed genes. We see that our hordeins are downregulated, so there's a B3 hordein, a B1, downregulated a lot. That's what we expected. So, like I said, there's all these other genes that are regulated as well, but we're just focused on these hordein genes. So I went to the genome, and anything that had anything related to a seed storage protein, a hordein, a gliadin, anything, we got it's probably about 250 genes that were predicted to be a seed storage protein, and we got all their expression numbers, so they don't have to be differentially expressed. And yes, we see lots of downregulation, so some of these might not be considered differentially expressed, but they are downregulated. One thing that we found was interesting is you do see compensation. So here's a gamma-3 hordein. That's going up. It's going up here. You see D hordeins. We mentioned that they didn't follow the same pattern in methylation. Well, they actually go up quite a lot in expression. So we see some compensation. If you look at the beta-amylase gene family, like I mentioned, we used to only have 2. Now we got all these other ones that are beta-amylase, which is interesting. And my question is, are they real? But we do see the beta-amylase 1 being largely reduced. We do see compensation, so BAMI2 is going up. We see a BAMI8 going up, so we see some compensation. But one thing that really struck us was this. Bowman, 8800, that's a high expressing wild type. But then what is this? 35,000? That's higher than our malting cultivar right here. So gene expression doesn't automatically mean enzyme activity, but we did find that, yeah, the enzyme activity was significantly higher than any other line we tested, and it's one of the higher ones I've ever seen, and I've been working on it for a while. And so here's the beta-amylase, or the Bowman Lys 3A. It's up at around 40. Here's where it's slooped down here. Even the wild type doesn't get to 30. So it's a really, really high beta-amylase line. Now, while we were in the middle of this, a couple papers came out that identified the causal mutation of Lys 3A. Turns out the Lys 3A mutant is a mutation in the barley prolamine binding factor. Prolamine is another term for seed storage protein, specifically hordein. So it's a transcription factor that turns on hordein genes. So now we got to go sequence our genotypes and make sure that we actually have this causal mutation. So Bowman Lys 3A, you see this T, it's got a mutant BPBF, right? Here's our Bowman Lys 3A, look at that. Where's our mutation? Okay, well, so apparently during backcrossing, they were selecting for a phenotype, which was a shrunken seed, and they must have backcrossed that mutant gene out of it because it has wild type. So what were some of our final conclusions? Well, these mutant lines that carry the wild type mutant gene, the wild type BPBF gene, right? They have similar patterns in gene expression, right? So we see increase in this one, it goes along the whole way. These B3 Hordeans, they're downregulated. You see that in all of these all these lyse 3A lines. However, some of them have the opposite, right? So even though Bowman lyse 3A has the wild type gene, it's having the opposite expression patterns, like in beta-amylase as well as in gliadin, right? So some of the genes, it's following the same regulation pattern, and there's some genes it's not. It's kind of confusing. But one thing we do know is that this transcription factor controls the regulation of all the Hordean genes in some regard and the beta-amylase 1. So if you look, this is between SLOOP and SLOOP lyse 3A, so we know this has the mutant transcription factor, and you just see a lowering of every gene. So this is a real-time PCR assay. So it reduces the expression in almost every time point. Over about 1,000 genes were shown to be regulated by this transcription factor. My hypothesis on why we still see some of the similar traits in the Bowman lyse 3A without having the causal mutation is that they use a chemical mutagen, so there's more than one mutation, probably a lot of mutations, and that even though it backcrossed 5 or 6 times, it retained some of those mutations that conferred some of the expression differences. But that's my only hypothesis. Kind of interesting though. So overall final conclusions, it doesn't seem like this gene affects global methylation because it did not really account for all the gene expression loss that we observed. However, we do think that this gene, this B1220 right here, might have been the candidate gene for the one described in the PNAS paper where they showed that the wild type endosperm was demethylated and the endosperm in the lyse 3A mutant was methylated. So they could have been looking at that specific gene, but it doesn't seem to be for all the Hordean genes. Increase in beta-glucosidase gene expression might be the cause for these low beta-glucan numbers. And then we found this really, really high beta-amylase line that I think is going to be, could be a good breeding resource for anybody that's looking for high beta-amylase. So maybe distillers or somebody looking for a really high BAMI line. Okay, that was one project. We're going to end on another project. So I mentioned earlier that we did the transcriptome for malting barley. So that's what we're going to end on. We use Conrad. So Conrad is essentially what the Malt Quality Lab at the USDA uses as their positive control. So if Conrad makes it through, then they know it was a good malting run. So we decided to use that as well. We used that system that I showed you earlier to malt it, and we sampled it daily, and then we did some analysis on it. So here's a picture of what all of the samples look like, the time points. So you got dry barley, and then this 0 DOG, days of germination. It just means that this just came out of steep, and then 24 hours in our germination chamber, another 24, etc. So when you look at all 21 comparisons, there's a lot of differentially expressed genes. It's a very upregulated process, which makes sense. It's essentially controlled germination, so it makes sense that you're getting a lot of increase in gene expression. When you look at the transcriptomes from each individual time point, you see that they kind of segregate in certain areas. So the dry, obviously, it's way up here, 0 and 1. So these 3 time points kind of very different, whereas these 4 time points kind of lump together. And we'll see that when we look at it even further. So 4 stages we identified based on expression patterns. The first stage we called stage 1, and that is essentially germination. So the physiological definition of germination is like the radical protruding from the barley grain or the grain, and that happens out of steep. So physiologically, it has germinated. In the malting lingo, it's still got germination to go. But so you see a lot of downregulation. So you see 50% are upregulated, the other are downregulated. So all these vestigial transcripts that were left in the developing grain, they start to become degraded and used for the growing barley. And the other half are the ones that are important for germination. The second stage happens between 0 and 2. So right around here, and you just see this large increase in gene expression. So you see a lot of genes being upregulated, a lot of gene ontology categories considered enriched. And then after that, we see kind of the shift downward in gene expression. So more genes are being downregulated. And that ends with not much going on for the last 4 days. So you remember where those in the principal component analysis, you saw in the upper corner all those 4 time points. Well, that's right there. You just don't see much change in gene expression during these time points. All right, so this is a gene ontology tree. So each one of these differentially expressed genes have these metadata attached to them that tell you what they do. If it's a transcription factor, they'd say that. Hydrolase is something that beta-amylase would have. So when you have differentially expressed genes, they can be lumped into categories, and if they lump into a category that's highlighted, that means this GO has been enriched. We're only going to look at this one. This is hydrolase activity. This is where my DP, diastatic power enzyme genes are. So from that box, we have 129 differentially expressed genes from the hydrolase activity. The malting quality parameters that we are over here in the red. So beta-glucan will be here. I won't talk about them, but you'll see all the genes that go up and down. Here's our diastatic power and alpha-amylase activity malting quality traits, and here are the genes that get changed there. So we see a lot of action in the AMI. These are a lot of genes going up. BAMI, this is the one, BAMI1 is a gene that gets turned on during development and does not get activated during malting. So this is not uncommon. All right, so what we were looking at here was essentially malting, the whole malting. So going from dry to 5. So those are the 2 pairwise comparisons. Steeping, which is essentially all you do is imbibe the seed until you get 45%, which is germination. You see a lot of downregulation and upregulation, but here's our hydrolase activity. And you see a lot of genes being downregulated from dry to 0. However, you do see some upregulation in some of the alpha-glucosase genes. So a lot of downregulation in the DP genes during steeping. However, if you look at the early germination, so 0 to 2, where you have a lot of those genes being turned up and down, you can see the GO tree got a lot larger, a lot more categories being upregulated. And there's our hydrolase. We'll look at that. And here they are. Look at all of the AMIs are going up, alpha-glucosase going up, one BAMI going down. But otherwise, all these DP enzymes are going up, which makes sense because they're being expressed. They're making enzymes, which makes sense because we use those enzymes to make our fermentable sugars in the next step. So that last stage where you don't see much going on from here, one thing I find interesting is between 2 and 5 DOG, you see this response to biotic stimulus going up. And what that tells me is something's growing on there, on the outside of it, and the plant's responding to it. And we know that there's a microbiome in us, and now we know more about microbiomes in plants. And so there's something microbiome going on during malting that could be beneficial to learn more about. All right, so now those were all differentially expressed genes. I'm just interested in the diastatic power enzyme genes. It's great if they're differentially expressed, but I want to know everything about these genes, and I know the ones that there's some that we know. So we know that AMI1, AMI2, we know BAMI1 and BAMI2, we know AGL1, but we don't know about all these other ones. We got the genome sequenced, so I went in, I pulled all the genes that could be alpha amylase, that could be beta amylase, and I graphed them out. So my favorite gene family, the beta amylase gene family. So down here we see enzyme activity going up, which is what we know. BAMI1 is considered endosperm specific. It's made in development, and it's activated during malting. BAMI2 is a more thermostable beta amylase, but it is not as highly expressed. You see it down here. But it does go up during malting. And then the most interesting thing is, see, these are all the genes that were differentially expressed. Here's how high they were expressed. This one right here, BAMI3, very, very highly expressed. It's actually higher, more transcript than BAMI1 in dry seed, and it actually goes up again during malting, and it ends much higher than any other beta amylase. this is very exciting to us. We don't know if this encodes an actual enzyme yet, but we should know by the end of this year. I mentioned earlier b-amylase. So when I was in grad school, beta-amylase was not expressed during malting. That was known. It was in the textbooks. Beta-amylase was expressed during development. Well, b-amylase we found does get turned on during malting, and then in that previous slide we see b-amylase gets turned on during malting. So we're learning some stuff about enzymes that some people thought were already figured out. So that's kind of cool. Alpha amylase gene, like I mentioned, AMY1 encodes a high PI amylase. That's been highly studied. AMY2 encodes a low PI. However, there's all these genes. We knew 2 of them, but now we know like 12 of them. A couple of ones that we're interested in, especially this alpha 2-2, it's low in the pantheon of alpha amylases, but it's still higher than any BAMI that we measured. So the one reason we're interested in is because it's novel, and it hasn't been published on, so we don't really know much about it. Another reason is the Molly in 2018 did the proteome of malting barley. He also found this one. So we know it's actually being made into a protein. So we want to look into that one a little further. Here's our alpha-glucosidase gene family. So there are 12 AGL genes. This is the one that everyone knows about. And it actually didn't even register on the differentially expressed genes. You see a little bump, though. It does go up. It's pretty highly expressed. But what is this one? That one has not been discovered. That has not been looked at. We don't know if it actually creates an enzyme that is functional, but we do know it goes up quite a lot during malting. So we're very excited about studying that one. Debranching genes, well, there's only one debranching gene that we know about, and that's limit dextranase. I did not find any other ones in the genome. However, there are these isoamylases that were found, and we've only studied a few of them. So this one right here, 1-2, has not been looked into, but we see that it goes up quite nicely during malting. However, isoamylase has only been implicated in starch synthesis, not so much starch degradation. So we don't know if this is going up because it's like a germination-related gene, or is it actually, could it have some implication in starch degradation? So we want to look into that a little further. Oh, yeah, that was the one exciting. I put in a lot of animations in the last few weeks. So these are kind of the genes that that we ended up deciding to look into further. We've got antibodies for these, just got in the freezer. I got this gene being made right now. I should get it in about 2 weeks. So we're pretty excited about that part of our research. Another way that we're going to study all these genes goes into my last part of my talk, the few minutes there that I want to talk about, is this collaboration that we have had with the WCIC, the Wisconsin Crop Innovation Center. So as a geneticist, not a breeder, but someone who likes to play with genes, it's always been tough on us working with barley because we don't have the resources that Arabidopsis have. So if we want to get a transgenic out of barley, we got to, 1, do it ourselves, and 2, don't mention it to our stakeholders, although now we can. Before, barley and transgenics, you couldn't say it in the same room. Although I just gave a talk on transgenics to the American Society of Brewing Chemists, so they're warming up on transgenics. But so we wanted a system, one, that we could use here in America, because there was only one system that you could purchase from, and that was using Golden Promise, is one cultivar using callus-based regeneration, and is from England. And let me tell you, getting transgenics from England takes a long time. Got a lot of paperwork. So we wanted something here, local, and so we got an agreement with WCIC, and we wanted to develop a system using mature embryos, or the meristem, to regenerate the plant, as opposed to using callus. So when you regenerate from callus, which is a bunch of undifferentiated cells, you can get some genomic issues when you regenerate. So you can get some epigenetic issues, and essentially, if you want malt quality, you don't want the genome to be weirded out. So we wanted something that was a little more resistant to that, and we wanted to use these meristems. And our hypothesis was that we could just use any cultivar that we want, like a malting cultivar that's being grown in North Dakota right now. We could use that as our target, and so potentially you could still have some economically viable barley when you're done with it. A little history on what the meristematic transformation is. So essentially this is what Monsanto does. Now it's called Bayer. But Wisconsin Crop Innovation Center was essentially Monsanto that was donated to the university, and they became this. So a lot of the intellectual property guys, so the smart guys that did this for Monsanto, they stayed, especially Ed Williams, who was left over from Monsanto, but he did all the work on this stuff. So here is our barley mature embryo. Here's what it looks like. So he had to remove all of these and find an efficient way to remove all of those. I don't know if he ever really found the most efficient way, because I know he was doing it by hand for a long time. But he got a whole bunch of these, and we were able to test the system and get it up and running. So there are two ways you can get DNA into your barley in this example, and you can do it biolistically, shooting DNA in, and then you could use the agro and manipulate that. So first step, can we actually get transgenic expression in these mature embryos? Glowing blue, or these blue lines, means yes, there's our transgene. So you can shoot, or you can use agro, you can get the DNA into these meristems, and we can get expression. We were able to then identify a good selective agent, so you put in a gene that's resistant to hygromycin or something, and then you grow it up. We identified a good selective agent. Then we needed to know, can we actually regenerate a full barley plant from just those embryos? Yes, we can regenerate a nice healthy barley plant from these meristems. Now, so it's transiently expressed, but when they grow into a plant, is it going to get into the leaves? Is it going to get into the germ? We determined that here's one event. 13 out of 13 leaves had GUS expressed. Not the best pictures, but they are blue. So that's positive. Another test we found, 23 out of 24, if you just looked at PCR, and then the GUS was expressed in 20 to 24 leaves. So yeah, we're able to get it stably expressed in the T0 plant. Now, will that plant put that transgene into the seed? So there are two layers of the apical meristem, L1 and L2. So in order to get it into the seed, you got to get it into the L2. So what we found was we grew those plants up that you just saw, took those seeds, germinated them. We got some good germination, so the seeds are viable, so that's good. Then we grew those seeds up, checked the leaves for positive GUS expression, and that's what we found. So we found that this protocol was able to get transgenic barley using mature embryos in an elite line. And we used something called GemCraft, which is a malting cultivar, more for craft maltsters, not so much for like adjunct based. So success. Well, what did we get out of it as a USDA facility? So we got a transformation service that's now offered at the WCIC, which is very exciting. The three USDA scientists that were a part of this got some transgenics to play with. Dr. Brigetzer got some transgenics with potential fusarium head blight resistance. My boss got some transgenics that might have some drought tolerance, and then I knocked out all the beta-amylases. And I got another mutant too, but this one goes along a little with this talk. So we're pretty excited about that. I'm over my time now, so we don't need to do that. But got to thank everybody that had a part of this research. Carl Simmons did a lot of the methylation issue problems. So he's a rock star. He's down at Wash U in a lab. She was here, Zhang lab, and then she went down to Washington. But he was phenomenal. David is my new technician. So back in February, I had nobody in my lab. It was just me. Now we have David. Got Sarah's here, so we just got a couple new hires, so we're excited about that. My former boss, Cynthia, and she was also my PI during my graduate studies. Molly, Jason, Sarah, the PIs, these guys keep our building running. All of our collaborators are huge. Corey and these two guys did a lot of that transcriptome work, and these guys did all of our malting. And no barley, no beer, right? Thank you very much. This is me, what I do outside of the lab. So thank you guys for listening, and I can take any questions if you guys have.
And if you wait a second, the microphone. Yeah. Any questions?
Yeah, I was just curious. What is the purpose of the helium you mentioned?
Oh, the helium?
Processes? Yeah.
Yeah, well, I can't get that anymore, can you? The helium is used to essentially build up pressure when you're doing the bioballistics. So you have this little rupture cap that will break under a certain pressure. So how hard the gold and the DNA will get shot into your tissue. So you turn on the valve, helium builds up till it gets to a certain pressure. Then you hit a button and it releases it, kind of like a gun, and it shoots the DNA through that rupture disc into your tissue.
Could you use other rare gases if you wanted to?
You know, if I wanted to resurrect that research, because I did some transient expression where I got that glowing barley picture, so I've done a little of that, and I don't know if I could find helium now. It's kind of tough to get. I know that GCs might be using hydrogen. I don't know if I want to use that, but I'm wondering what people are using. I don't know, but they're going to need something else.
Why is there a shortage of helium then?
Well, there's only, from my knowledge, this is definitely not the end-all be-all, but there's only a few places you can get helium from. I think one in Russia, one in Texas, and like every big lab, every medical device uses helium, I think, to cool things, and so it's really common in medical usage and then in research labs as well.
So you mentioned, I got three questions.
Okay.
Two are practical. Why is high protein bad for beer? Maybe naive question, I don't know.
Well, high protein can affect filtering. It can affect the brewing process, right? And also you can get unwanted things in there, so it can kind of gum up the brewing process. Yeah, so that's part of the reason.
Does nothing to do with taste, right?
Well, protein is very important into taste. It's actually really important for foam stability. So like a lot of these hordeins and beta-amylase you'll find in your foam. So it is important for that, but then certain proteins can kill your foam. But from my understanding, I think it messes with the brewing process downstream.
Okay, and in any of your mutants, did you ever make a beer out of any of them?
I wish. That would be interesting. Yeah, well, I have a couple. So I have a high thermostability line transgenic that I do want to make beer with. So I've done more of the beer making in my driveway, not so much in the lab yet.
So I got one extra one.
Okay.
So these transgenic plants, do you have to worry about getting them released? That would be a big controversy.
Yeah. Oh, you have to write protocols on what you're going to do, how you dispose of them, where you're going to put them. We had to put signs up on the door that says, authorized personnel only. Yeah, no, they really want to know everything about them. I had to get the plants over here, to get the seeds over here, I had to learn more about the CRISPR than I knew. I mean, we had to find every piece of DNA that they use in the CRISPR constructs. You had to detail the origin, the base pairs, the sequence, and give that to APHIS to get. Took months. Back and forth. Yeah. So they definitely keep track of it. Someone does.
So a comment and then a question. You probably don't know that the person who found DNA methylation mammalian cells worked over here at McCardle Lab. His name was Tom Snyder. He worked with Ann Potter.
Okay.
He was my grad advisor.
Oh, right. That's excellent.
So
I love it.
Weird, but that's a UW history story.
Oh, that's great.
It's a 1965 or 66 paper.
Oh, wow.
On methylation in rat hepatocyte cells.
I love it.
But because I'm a geek, the demethylation process, is that the same as the TET process in humans? TET genes?
What? No, I don't know if I'm familiar with the human TET, but I know I'm assuming some of it's similar. There's a lot. So there's a different. So in this methylation, it's more of like allowing transcriptional machinery to get going. But I know that there's methylation on histones. There's a lot of different methylation that can affect humans as well as plants. And like I said, the callus-based regeneration can give you a screwed up plant because it alters all those the methylation. And I think that that mutant that I mentioned is a lot more convoluted than I talked about, because if you breed it with certain lines, it gets really weird segregation patterns, and it's so that loci is located near a male sterile gamete gene. So I don't know. It's got some interesting papers out there on it that it doesn't play nice with people sometimes.
I'm a little bit curious about in which extent could you silence the gliadin gene expression to produce something less toxic for the ones who have celiac disease? Yeah,
so that is, I've actually been thinking about that. So like the Australians that I worked with and I got some lines for, they did a traditional breeding to knock down the celiac, the gluten. The only problem with using it to knock down those gliadin genes, the Hordian genes, is that there's a lot of them. And if let's say, one thing we were talking about with WCSE is that if you target the promoter region, like the minus 300 element, which is a cis motif, but then you're going to knock out everything or you could, because these Hordians are very repetitive, which is kind of why they cause us problems, because these repetitive regions, they break up and there's just a lot of them, so we create a response to them. But if you target one of those repetitive regions, then you might be able to put holes in a lot of these Hordian genes to create like a CRISPR-generated celiac barley, sensitive barley, something like that. It's definitely something I've thought about playing with for sure. Because I know I didn't drink beer for the last few years, but I started drinking again, and I do remember that sorghum beer is not very good. Millet beer is not. I know there are people out there listening that might like those beers, but a barley based beer that's gluten free, that's pretty desirable.
Will Rogers called to say he never met a beer he didn't like.
Oh, that's right. The best beer is the beer in your hand, right?
Yeah.
When looking up the etymology of some of the cool words in barley and malting, the genus name for barley is Hordium.
Hordium. Vulgare.
Common Hordium.
Yep.
And does Hordium come from bristly? And because that picture you showed of ransomware,
yeah,
that's a very heady, to put it, I don't know if that's the right agronomic word.
Well, it's got larger awns than wheat, for sure.
And are there any beardless barley?
Yeah.
Or
yeah.
do you
There's naked barley.
What's that?
There's naked barley.
And can you malt that?
Oh, they're trying. So Pat Hayes over in Oregon State has this, and I think there's some UW people too, working on naked barley. So I know they're using it a lot for baking and stuff, but they want to be able to brew it as well. The only problem with naked barley, no hulls or whatever, is that the hulls are actually very useful in the brewing. They end up creating a bed to filter the wort. Without that, you have to add rice hulls or something to do that, but I know that they are working on it for sure. Yeah.
And this picture is amazing to me because I think this is the first time I ever understood why they call it Hordium, because of all the bristles.
Yeah, I don't know the etymology behind the Hordium. I used to like the vulgare, right? Because common, that's what the vulgar, right? Swearing is what the common people do after they drink the beer. No.
And could you go to the pictures that you were showing at the end?
Oh, the end? Oh, yeah.
This is right at Tiny Park you mentioned, right?
This one?
No, the malt house there.
Yeah, well, that was I put that in today. I have to go read the book. It's on Lake Mendota.
Yeah.
I don't know exactly where.
Right at the locks.
Did it say that? Yeah, it might have been right there.
It's where the salmon swim in. At the
upstream.
Can you show those pictures again?
Of?
That you had it, and so are you doing a chemistry show at schools?
So yeah, it's more, well, so you can see I'm talking a little bit about malt and barley, so I like to show them all about barley and what we do. Obviously, I don't talk about beer very much with kids, and then I do a little chemistry, right? So I use liquid nitrogen because we use that to do our research, but then they like to. What am I doing? That's at the end. I dump a whole bunch of hot water and it creates this big explosion. And then I'm doing a rose. My middle daughter's middle name is Rose, and so I smash it for her. And then yeah, and I do some other things that aren't like, I think I do like anything with liquid nitrogen. I just do a bunch of fun. They liked it.
And in your right hand on the left picture, left my
Whoppers.
left. Whoppers. So
Yeah,
that's malted?
Malted, yeah, malted milk
Okay.
balls. That's right. So yeah, you can just go in your cupboard and like if your bread has more than 3 ingredients, a lot of times it'll have malted barley in it because it's called a bolted flour, right? They put a little malted barley in there to help it to germinate or to
And
rise.
what are the other two things there in the boxes, the yellow box and the red box?
Looks like muesli, and I don't know. So some of them were kind of like old. We had them sitting around the CCRU for like 25 years. So what I do is I took a handful of those, and then I take a handful of what I have in my cupboard at the time, and then I bring it. And then I have wild barleys, and you can see right here there's a black barley. This is a naked barley we just talked about. It's from Ethiopia. So yeah, we kind of show them that stuff, and then entice them with blowing up things.
Very nice. And what hockey rink is this?
Which one? That's Verona, Eagle's Nest. That's
Yeah.
in Sun Prairie.
Okay.
That might be in Reedsburg. That's in Middleton.
Okay.
And
I don't-
that's Kiva.
Good.
So yeah, I got my daughters. So yeah, so this is my oldest. This is the one that was getting picked up.
Okay.
And then she helped me coach my youngest, and then that's my middle. So
Very good.
keep me out of trouble.
Here we go.
Yeah, I was just curious, was that Carlsberg Laboratory in Denmark, was that related to the brewery over there?
Yeah, so it might be because a lot of these old breweries were very big into research, right? So I think there's a stats test. It might even be, I don't know if it's Fisher's, but there's a large, maybe the student's t-test, I think was a Guinness researcher. Like worked at the Guinness labs. And the correct stat might be eluding me, but I do know that one of the major statistical tests was found